Okay, let's have a class. First of all, I would say this. Let's do that. 9:30. Let's start probably 9:35, 9:40ish, because the traffic is really bad. I mean, sometimes I have to grab these things from my own lab. So let's just start 9:35, 9:40, 5-10 minutes delay. You can come here, get your results, and also get those material. Okay. Second thing I would love to say is that we are getting close to the semester, and exam 3 of the lecture, I just want to tell you everybody here, okay, exam 3 for lecture. This will be November 10th. Is that right? Based on the syllabi. Now we still gonna have a practice exam. So practice exam will be on eCampus over weekend. This will be over weekend. And then, as normal, we had 100 points in the exams and with some 8 to 10 points extra. That's normal. Then we have lots of extra points will be glycolysis, TCA cycle. This will be chemical, name, and structure. Okay, the chemical name 0.5 points each. Structure 1.0 PTS. Okay, points each. Then all side chain of amino acid. How many amino acids? Lots of people ask me how many amino acids? 20, is that right? Yeah, you should know. If you ask me how many, then I think you have a big problem. If you tell me 21, that's good. It's the new textbook. Some people are saying 21. We'll talk about it in the lecture. But if you don't even know there's 20 amino acids, they're not good at all. Okay, 20 amino acids. We're going to have a name, a full name or acronym, both are fine. Okay, and the all side chain. This together, 1.0 points. So how many points extra? You go ahead and count it. About like maybe 50-ish. Okay, that's more than enough for you to make up what you're missing. Okay, so that will be the plan. Now, November the 8th is general election day for midterm election, so we don't have a class. And the next Thursday, we will be doing a review. So you almost have a week to revise. Or to review and do. I will suggest you have a study group. If you don't know each other, using this lab like a bench partner, 4 of you, 3 of you, study together, then to prepare exam 3. Because this is pretty much the last opportunity to catch up if you're missing some grades. And I hope you to do really well. I'm not cheating you. It's going to be exactly the same. I left 3 empty pages. Glycolysis, TCA, and amino acids. I'm not saying pick one of them. It's going to be there. I'm not joking. I'm not saying you prepare all like 3 weeks. So finally said, I only tested your one side chain of glycolysis. It's going to be there. I just tell you, 3 empty pages are left will be for you to do. Okay? So that's what we talk about the lecture exam. Okay, lab, pretty much what we're getting close also. Today we will do a pond water analysis of APC and coliform. Thursday, next week, Tuesday, we will do is the eastern modes analysis in a very molded sandwich. I already made that. Very molded is in my Windows aisle of my office. So I'm gonna bring it. We're gonna do that. Then next week, Thursday, we'll do the antibiotic resistant test. Following, we will do a analysis of bacteria in the food products. We have a different food products. Then we are going to do the review. Then that's all. So we're getting close to the lab section too. And remember, and the lab usually finish earlier before the lecture. So lab exam will be the Tuesday after Thanksgiving holiday. Okay, it will be November 29th-ish if you look at the calendar, that week. And I said there is a 20 points, extra points, which is your lab notebook record. So please do that. If you haven't done yet or you need it. Okay, it's like your insurance. Bring you lab notebook and we are gonna grade it. and then your final grades we put on the e-campus. And before you go for final exam, and all your attendance point, the homework, all the exam ones, they are all on the e-campus. So you get to know how it goes. Okay? So these are the left only final exam and what you needed for the extra points of the lab notebook. Okay, so that will be the plan. So today we will do microbiome analysis of pond water. And we are using a very special media. So first of all, we want to talk about indicator microorganism. So what is indicator microorganism? Basically is we have some general standards. Some of the microorganisms we're going to test in a sample, usually water sample, food sample, we will know the total hygiene situation. Said very simple, how clean it is and how dirty it is. So what are we going to do is number one is APCs, is aerobic plate counts. This is basically is how many bacteria, total bacteria, which is how many total aerobic bacteria. Very special. We not use media. Of course, you can do on the agar media using TSA, nutrient blocks, nutrient agar. We're going to do in these special, coming from a company called 3M, so we call it a 3M petrofilm, which is a paper media. All the bacterial ingredients is in this piece of paper. So it's very magic, okay? So we're going to do is a 3M petrofilm. Now, there is no standards for how many aerobic plate counts in the food products. But usually we say, as a normal products, let's say a beef or meat products, and usually we say the normal background microorganism is about 3 log 10 CFU per gram. So this is normal. And most of the time, if we see APC is more than 5 log 10 CFU per ml per square centimeter or maybe per gram, that is not good. Which means you basically is failed standard sanitation operation procedure. Okay, you will say this is we call it's a failed SSOP. So we don't have a really legal standards, but usually 5 log is a top limit. The second thing, this is the first thing. The second thing is we're gonna look for is coliform and E. coli. The same thing we use a petrofilm. It is a little bit of difference. The petrofilm looks like this. There is a big circle in the center, and it's a pink color, and this is APC petrofilm. Can you see it a little bit difference? The same thing, the media is all on the center. So what we are looking for? These two basically is intestinal bacteria. So ideally, in the food products, in a pond water, or your drinking water, you should not have E. coli and coliform. And this E. coli is generic E. coli. Now, what the standards for these, what they are looking for? They are looking for is fecal contamination. You absolutely shouldn't have E. coli. In the United States, the city drinking water system or municipal water system, there is an EPA regulation, Environmental Protection Agency is 0 tolerance for coliform, E. coli, generic E. coli, and even fecal coliform. So what comes out about these things? Long time ago, people are using coliform to do a standards. If there's one coliform there, which means it's not a pass hygiene standards. If there's a no, which means you pass. But later on, people find to see the fecal contamination, coliform may not be a good indicator. We needed to using generic E. coli. Then generic E. coli become a better one. At the same time, sometimes people also test the fecal coliform. Now, what is the difference? It is the only difference is the incubation. Fecal coliform, the incubation time is 44.5 degrees Celsius, 48 hours. It is the standards. Coliform and E. coli, it is only 35 to 37 degrees Celsius at 24 hours. Okay, that's a major difference between that. So today, we are going to test how many APC, Arabic plate counts, and E. coli and coliform in this pound of water. This is coming from the river close to the Morgantown, close to WVU. Okay, just a pound of water. Obviously, there's lots of bacteria there. Because it's not going through filtration system. There's no colonization system. It's not drinkable. And also you need to know pound of water is hot water. Because calcium and magnesium is higher level. Okay, so we're going to do. How we do it? We will get is a pound of water. Let's just say this is a pound of water bottle. First thing first is you have to shake. There is no way you go there just to grab something to do. First thing first, shake. Usually better 15 seconds, back and forth, at least. Okay, before you do, all the liquid, you need to have the habits, which is shake, because it's not uniform. So usually we shake at least 15 to 30 seconds. Second thing, dilution. We will do also 100-fold dilution. So how to do it? We talk about this all the time. You have 3 tubes. Okay? 3 9.9 ml tube, each containing 0.1% nutrient loss, buffer solution for bacteria. So how we do it? Using pipettes. This will be 100 microliter pipettes. Adding into the first one, mix very well. Then taking 100 microliter. This means 0.1 ml, you know that. Okay, then go second one, and then third one, 0.1 ml. Now what is the dilution factor of the tube? Is 10 to the 2, 10 to the 4, and 10 to the 6. Is that correct? Yes. Okay, now next one we want to very important to emphasize. Petrofume is different from the agar. How many volume we gonna add? Is 1,000 microliter. That is 1 ml. It is not 0.1 ml. Okay, so therefore, it ends up with, in the petrofume, the tube dilution and the final dilution are exactly the same. So we're going to label on the top is your name, batch number, and your dilution. That will be first one 10 to the 2, 10 to the 4, and 10 to the 6. Okay, that's exactly the same. You're going to do this for what we will to do. We will do all those 3 for APCS. APCS is right here. I don't know how many you have. It should be good. Everybody gets 3 pieces. For the coliform E. coli, we get 2, everybody. Okay? For coliform and E. coli, we will do we usually call it ECC and TCC. Everybody get 2 pieces. This is you get 3 pieces. We have enough, so you're gonna do it. Okay? Now, after doing this, what we're going to do is incubation. 35 degrees Celsius for 24, 48 hours. And anyway, we're going to do next week. Now, here is the difference. We want to mention about the results. Okay, APCs. What did APC looks like? APC, after incubation, you will see all those pink to red dots. Colony. And then you just count it. That's simple. The tricky thing is this guy. Is this E. coli coliform petrofilm. What's the tricky thing? E. coli coliform. So the media looks like this. So you're the one right in the center. What are you going to see it? You will see majority of them are red. This is coliform. No question. Then you will see some of them is a little bit blue. And then it's like a gas bubble. This is E. coli. Then what is the total coliform? Add up together is the total coliform population. This is the key. Lots of people doesn't know this. You have to, if the results here, people ask you, how many E. coli are there? You count only blue colony with gas bubble. Now, it may not have it. Okay, that's the thing, because it's near the campus, you may not have it. Or maybe 1 or 2. Coliform is also the red dots. Now, when we say total coliform, you have to add up them together. Okay, so the total coliform, the acronym, basically we call it TCC. So that's petrofilm. We usually call TCC, ECC, TCC petrofilm. ECC is E. coli, TCC is total coliform. Okay? So that's a procedure what we're going to do. So today's work is very simple. You already have is a 3 tube. You're going to come here to get, everybody get 3 these petrofilm. I need to find more. Maybe we need more. And this is everybody get 2. And I would love to show you some technique how we're going to do this. So I use one as an example. So everybody come here to the desk right here. Okay. I'm going to show you. This is the key. Okay. Can I borrow gloves? Do you have gloves? Yeah. I'll just borrow one more. Thank you. Very quick, just to show you how to do it. Dilution, I will just skip it. Okay, I will just skip the dilution because you know how to do. Same thing like you did before. The key thing is that the label. The label is always on the top. So you label CS, bench number 1, and this is, let's say, 10 to the 2, for example. And this is on the top, we label CS, bench number, my initial, bench number 1, 2. Okay. Let's say this is a dilution. Give me one more tube right there. Grab me a tube there. Just as an example. Let's say this is the one you finished the dilution. Okay? Put it here. Can I have a tips? And what we're going to do, we are not going to using 100 microliter. This, you need to adjust to 1,000. Because add on the petrofilm is always 1 ml, is not 100 microliter, not 0.1 ml. This is the key. So if you see today, you bench partner or yourself, one tiny drop on the middle, it's wrong. It needs to be a big drop. Okay, so make sure after you finish dilution, it is at 1000. Then what you do? It's not difficult at all. For APC, let's say we all finished. Have your hands on right here, very gentle at the middle. Cover it. Use a presser to gently press it. That's all. You need using is a rough one, not a flat one. Okay? If you use a flat one, you press, all comes out. So what it looks like, you will see a big circle in the middle. You will see it. Now for this guy, what you gonna do? Same thing, you finished. Absorb 1 ml. And then, very gentle to take it out. Right in the middle of the circle. Okay, try slow, no bubble. Then, very gentle. That's all. It will be gradually cover the whole surface. You don't have to use a presser because there is an edge of the well and eventually it will be all covered. So, later on, do you have a container here? Later on, it will be pretty much like, no, it's off right now, but it's pretty much like gonna be cover the whole area. Okay, it's gonna be like that. This you don't need a presser. The others are the same. When you do a dilution, you need to change tips. So we still need 100 microliter tips. Yeah. And when you add to the final, is also from higher to lowest dilution, only 1 tips. And you also look at the dilution factor. You do 10 to the 6, 10 to the 4, and 10 to the 2. When you go for 10 to the 4 to 10 to the 2, you have two different type of the petrofilm you can do at the same time. Okay, so it's not difficult. Everybody have 5 pieces of the samples. Okay, so that's all I have.